Hi guys, and welcome back on our channel, and this is our third video about the cardiovascular system pharmacology. In this video we're going to be covering the diuretics. Now first of all, let me ask you, well, whenever there is someone around you who was actually just diagnosed with high blood pressure, what's the first thing that would come to your mind is actually advising him or her not to eat salty foods, right? And on the other hand, if someone felt so tired and dizzy, and then you measured their blood pressure and you figured out that their blood pressure is low, you would recommend them to eat salty foods and sometimes the sour pickles, right? Now, so why do we actually ask patients not to eat salty foods if they have hypertension, and on the other hand we recommend eating salty foods when they have hypotension, right? This is basically because the water is always following the salt. Wherever the salt is going, the water is gonna go. So when I say salts, we actually mean mostly sodium or mainly sodium, right? And yeah, you can say sodium chloride, but it is mainly sodium. Now, let's actually just go over the physiology of the nephron and how does it really work. You know that in the kidney we have the cortex and we have the medulla. Now, the nephron is going to be like lying in between these two guys, the cortex and the medulla. So some parts of the nephron are going to be above the line dividing the cortex and the medulla, and other parts are going to be in the medulla. Okay? So we know that we have here the Bowman's capsule, which is going to be surrounding the glomerulus, where the filtration starts, and then we have the proximal convoluted tubule. Then we're going to go to the loop of Henle, and the descending loop of Henle, the ascending loop of Henle, and then the thick ascending Henle, and then we're going to enter to the distal convoluted tubule to the collecting duct. And the distal convoluted tubule actually has a proximal part and a distal part. Now, before starting with this, I want you to write this down, that all of the diuretics that we're going to speak about are primarily salicylic. Salicylic means that they are getting rid of the salts, and secondarily to that, the water is going to be lost. So they are basically inhibiting the absorption of the salts, and because of that, we're going to lose the water. Okay? This is applicable for all of them except the osmotic diuretics. We're gonna go in details in each type of the diuretics. Now, in the proximal convoluted tubule, most of the absorption is happening. Absorption of the glucose, absorption of amino acids, absorption of salts, absorption of the water. So most of the things are reabsorbed in the proximal convoluted tubule. Okay? In the loop of Henle, in the descending loop of Henle, there is a passive absorption of sodium. So, if the osmotic pressure surrounding the descending loop of Henle is high, water is going to move from the loop of Henle to the medulla, which actually means that we are reabsorbing the water. And if the osmotic pressure wasn't high enough, we're going to lose the water. Okay? Now, the ascending loop of Henle, on the other hand, isn't permeable to water. So it doesn't matter how much the osmotic pressure is here, water is not going to move across the wall of the ascending loop of Henle. Okay? Now, in the thick ascending loop of Henle, we have a couple of transporters that are responsible for creating the osmotic pressure in the medulla that is going to be responsible for driving the passive absorption of the water. This is basically a transporter that is absorbing sodium and potassium and 2 chloride ions at once. So every time it's working, it actually absorbs 2 chlorides and 1 potassium and 1 sodium ions to the medulla. Now, let's imagine that these guys are working very well. This means that these ions are going to be accumulating in the medulla. When they are accumulating in the medulla, what is going to happen here is that the osmotic pressure here is going to be increasing. And if the osmotic pressure is increasing, this means that the water is going to passively move from the descending loop of Henle to the medulla, which means that we are absorbing the water. Okay? Now, if we stopped this, somehow inhibited this process from going on, what is going to happen is that these ions are going to diminish gradually. So we're going to lose the osmotic pressure that should be accumulating here, and this means that the water absorption is going to stop as well. Okay, and the osmotic pressure is the so-called the corticomedullary gradient. Corticomedullary gradient means that the concentration around the loop of Henle is actually increasing from the cortex towards the medulla, and that's why the concentration of the urine inside the tubule is going to be the highest here. Why? Because most of the water is going to be absorbed, and you know, if you are absorbing the water from the solution, this means that the concentration of the solution is increasing. So the highest osmolarity and the highest concentration of the urine is going to be in the loop of Henle, in the bottom of the loop of Henle. Okay? Now, in the distal convoluted tubule, in the proximal part, we have also a sodium chloride symporter, which means that it is driving both ions in the same way, symporter. Okay? Sodium and chloride together. And at the end part of the distal convoluted tubule, we have a transporter that is actually absorbing the sodium from the urine, and on the other hand, it is giving a proton to the urine. Okay, so it is eliminating a proton on the cost of absorbing a sodium, or vice versa. Anyway, in the collecting duct, we can actually absorb water passively only if we had ADH working. So if the ADH is being produced from our pituitary gland, then the collecting duct is going to be permeable for water by a transporter called aquaporin. Okay, so now the sodium and the bicarbonate are going to reach the nephron through the filtration, and they are going to swim along the proximal convoluted tubule. Now, we know that whenever whenever it's an ion, this means that it has a charge, and it can't cross the membrane of the cell because it is lipophobic, because it has a charge. Now, the sodium will be absorbed because it has the transporters and they can carry the charged sodium and reabsorb it. What about the bicarbonate? We need to reabsorb the bicarbonate, right, to maintain the pH of our body. So what we're going to do is basically that the bicarbonate is going to combine with a proton and form the carbonic acid. Now this carbonic acid is going to dissociate into water and carbon dioxide. But how does this happen? Well, the association of these can just happen simultaneously. I mean, right away it is going to happen, because it is like, for example, saying that a sodium ion, okay, now when you actually write like a sodium ion has met a chloride ion and formed sodium chloride, does this need an enzyme to initiate this process? Not really. And the same happens here. So whenever the bicarbonate meets a proton, it is going to form a carbonic acid. Okay? Now, but the carbonic acid wouldn't dissociate normally into water and carbon dioxide, but it would dissociate back into bicarbonate and the proton. And here comes the function of the carbonic anhydrase. Now, so the carbonic anhydrase enzyme is the enzyme that is going to enhance the formation of water and carbon dioxide from H2CO3. Okay? Now these guys are going to dissociate, are going to travel through the membrane of the proximal convoluted tubule cells to the intracellular area. Here, a reverse process is going to happen. So these guys are going to combine back into the carbonic acid again by the function of the carbonic anhydrase. And then the H2CO3 is going to dissociate into proton and bicarbonate. The proton is going to be pumped back into the urine. We don't want it. We don't need it. So we pump it back to keep the urine pH maintained. On the other hand, we need the bicarbonate. So what we're going to do is absorbing the bicarbonate along with the sodium by a symporter in the basolateral membrane of the cell. Now, let's cover the types of the diuretics we have. We have two types of the diuretics that are going to lead to losing potassium. We're going to lose the potassium whenever we use these two types of diuretics. These are the loop diuretics that are going to inhibit this process here. What does this mean if we inhibit this process? We say that this process was responsible for the formation of the osmotic pressure that is going to drive the passive reabsorption of the water. So if we stop this process from happening, the water is not going to be reabsorbed passively here, and we're just going to lose it along the nephron to the urine. Now, this is the so-called the loop diuretics. Now, the second type of the potassium-losing diuretics are the so-called the thiazide diuretics. These guys are going to block this transporter and inhibit the sodium chloride reabsorption. We said that the water is following the salts, right? So if this was functioning, the water is going to be also reabsorbed after absorbing the sodium. Now, if we inhibit this, this means that the water wouldn't move, wouldn't leave the nephron, right? So we're going to lose it along the nephron again as a urine, and this is the function of the diuretic. We want to lose as much urine as possible. Now, the potassium-sparing diuretics, on the other hand, we have two other types. The first one is the so-called the aldosterone inhibitors. They are going to inhibit the transporter that is responsible for sodium absorption that is dependent on aldosterone, which means that this transporter works only if aldosterone is present. Now, if we block the aldosterone, this actually means that we're going to block this receptor, and this is going to inhibit the sodium absorption, so we're going to lose the sodium, primarily salicylic, secondarily diuretic. We're going to lose the salts first, and then we're going to lose the water. This was the aldosterone inhibitors diuretics. Then we have the sodium channel blockers diuretics, another type of potassium-sparing diuretics, and this one is going to be blocking the sodium channels on the apical side of the distal convoluted tubule. We're going to talk about that in details. Now, the carbonic anhydrase inhibitor diuretics are basically the guys that are going to inhibit the carbonic anhydrase. Let's imagine that we inhibited this carbonic anhydrase process. This means that the bicarbonate is not going to end up forming H2O and carbonic dioxide, but it is going to combine with the sodium. So it would actually form a sodium bicarbonate, right? And this is going to be lost in the urine, and this is going to drive the water with it. So we would lose the sodium bicarbonate together with the water, and this is the function of the diuretic again, that we're losing the water with the urine. This is the carbonic anhydrase inhibitors. And then we have the osmotic diuretics. Just leave it for when we talk about it in details.